Good afternoon and welcome. It's my distinct pleasure to introduce Dr. Xixing Gua today. He's an applicant for our informatics post in the Center for Precision Medicine Research. So a little bit about Xixing, that he received his PhD at Fudan University in Shanghai. So for most of you who don't know, Fudan University in Shanghai is China's premier university for genetic research. It was made famous by Dr. C.C. Tan. Dr. Tan was really a world famous geneticist who worked on moth and butterfly genetics with Dr. Morgan. So so this is one of the champions of genetics. And Xixing was actually trained by people who Dr. Tan trained. So it's a long line of fantastic training. You'll see that Dr. Gua is very passionate about his research, and he is also what you'll also find is his originality in thinking. He's an accomplished scientist, both in the laboratory, in computation, and also in genetics. And when we talk about genetics, I think most of us think about just DNA, right? AGTC, that's it. That's the blueprint. But it turns out that it's not all that simple, that there are modifications to DNA and modifications to the proteins, the chromatin, that are part of our genes. And those modifications play an important role in our development and in the etiology of disease. So Dr. Gua will talk today about his approach, both using computational methods, genetic methods, and what we call epigenetics, that is the non-DNA part of our inherited traits. And to use those to understand disease and where these diseases come from, and how we can address that to improve diagnostics and treatment in the realm of precision medicine. So, Dr. Gua, please.
Thanks so much for Dr. Burney's introduction. I'm really appreciative to have this opportunity to give the presentation to share what I have done in the past 10 years. So the title is Computational Biology, Genetics, and Epigenetics to understand what is disease and how to improve precision medicine. So for the whole project, we will have this outline. First, I will talk about what is human complex disease and what is precision, in my opinion. And another one is I will introduce the two projects for me to identify disease gene in genetic method. And for genetic method part, I will to identify two genes, two disease genes. One is the blood cancer gene, and another gene is hemochromatosis genes. In the epigenetic project, I will introduce some project I conducted to identify cancer biomarkers so that we can diagnose cancer at early stage. So there are 4 cancers I have conducted. For example, the non-small cell lung cancer, blood cancer, liver cancer, blood cancer, and the last one is suppose we know it's cancer, it's okay, we just do something, what we need to do. But suppose a patient come here and we don't know what exactly is cancer, so how to infer from the cell-free DNA to which cancer this patient get. So in the last section, I will give some plans how to absorb, external fundings to support my independent research. This is, I will introduce 2 projects. One project is as a contribution of genetic variants in epigenetic factors to to human disease. And another one is the cell-free DNA methylation-based pan-cancer early diagnosis. So let me give a very short introduction to myself. So I finished my PhD in 2005 from Fudan University. Then so before this time, I get a scholarship to pay a visit to U.S. university. So I received an opportunity to go to University of Texas and stay there for 2 years to train for the epidemiology and epidemiology genetics training. So later I finished my PhD and I get a job position in University of California, San Diego. So in that lab, I did a lot of cell-free DNA methylation research. And from end of the 2017, I get the position in Marshfield Clinical Research Institute. I work together with Stephen Schulte. So in the past 10 years, I published 53 peer-reviewed paper and get 2 granted patents. Another one is under review. So for the first author publication, it's just like this way. This is only showed the first author, and the co-author, I didn't show it. So for the awards, several awards I received from after I come to US. So for example, 2014, 18, and 19. So this is a very brief report. So again, so let's check what's our understanding to human disease in the 21st century. So for human, we have almost 12,000 diseases. We include 80 different autoimmune diseases, 100 different cancers, and almost 7,000 rare diseases. But no matter what kinds of disease, they occur in different tissues, but we always can find the reason in cells or in the tissues. So in the cells, we know the DNA information will be transcribed to RNA and then translated to protein. Any loop, if something wrong happened, we will get some trace. Not only this, because among these very important elements, we also have lots of modification systems in our human genome. For example, DNA methylation. For RNA also have methylation. Protein also lots of modification, especially in the histones. So now we know that for any disease, there are lots of risk factors. For example, from the genetics, we have genetic risk factor, environmental risk factor, epigenetic factor, or the lifestyle or family history risk factors. And for the quite a long time, maybe this gene, this pathway work together, and then we will have a pathway. And with several years, maybe we will get some phenotype. And after the individual comes to the hospital, we will have lots of information. For example, from the EHR, we will have clinical phenotypes, CD9 lab tests, and lots of image data, for example, EEG, ECG, and some other images. So from all this information, we can understand a disease with more details. And actually, if you enroll all these diseases, you can have very good computation firm to understand disease, to diagnose them, to find better drugs. This is actually the way we do in the computation way. So the next one is, so what's precision medicine? So for me, actually, I cannot tell the difference between these different concepts. For example, precision medicine, personalized medicine, smart medicine, or smart hospital. I don't know actually what's the difference between them. But as a bioinformatician, this is what I'm thinking is precision medicine is automatic medicine, actually. So we want to build some tools, some pipeline to help the physician to do everything very smoothly, very quickly. So for me, I just like automatic medicine. So then another question is how to achieve precision medicine. So in my opinion, we have 2 ways. First way is I call the direct way. In this way, we need to understand each disease one by one with very deep knowledge. You just pick up any disease. For example, I like cancer. I want to research diabetes. So you can research the disease one by one and find the disease gene, find the pathway, and later you can find what exactly is disease. But there's actually another way I call the indirect way. So in the indirect way, actually I don't know what exactly is disease. So I just want to know, okay, this disease looks similar with another one. For example, me maybe looks very similar with you, not like this desk. So okay, this is a guy, not a desk. So similar way, if we get lots of data for the sample, we don't need to care about what exactly it is. We just need to find the most similar phenotype or disease with it. So then we can use similar drugs with similar therapy strategy. So in this, so I can show you why, just from the data we can do this. For example, we have lots of different data. This is genotyping data. This is RNA-seq. This is genome-wide methylation data. So if you want to find the guy where it is from, so you can just use the genotyping data. For example, this is from multiple clinical research data. So we have the PMRP data. And actually, if you plot the genome-wide SNP data, you can find, okay, this guy is from African, this guy from Asian, and our PMRP data is almost nearby the European data. This genotyping data can give us, okay, where this guy is from. So then, our human, we have almost 200 different tissues. So how to separate different tissues? It's very easy, actually. You just use genome-wide RNA-seq data. With genome-wide RNA-seq data, you can separate different tissues very clearly. For example, this is the liver, this is the muscle, brain. They're quite clear. So how about the methylation? Methylation actually can give you more useful, more powerful application. For example, all these samples is brain cancer samples. But if you plot with the genome-wide methylation data, you can find, okay, genome-wide methylation data can help you separate different brain cancer to different cell subtypes. So one by one, you use different markers, you can find different stories. That's why I don't need lots of clinical information. Just give me the data. I can prove lots of things. So then now we come to the first part is I give you some example how to identify genetic variants for human disease. The first one is linkage analysis to find the blood cancer genes. So this story is happened in 2012. And we come across a very big blood cancer family. In this family, we have 11 blood cancer patients. And you can find lots of individuals. They didn't show the blood cancer, but it's very high probability they get cancer. So then we are quite interested in this family. So we did a marker array data to check this 13 different individuals within this big family. And we identified very interesting candidate gene regions. It's located in chromosome 20, and the p-value is quite significant. The ROD value is come across larger than 4, so we think it's very significant. So because this region actually is very large, we need to know the detailed information about it in this region. So we use another targeted next generation sequencing technique to sequence all these regions and find the variants. Very interesting. We find 2 exome variants and 15 intronic and 15 intergenic and 4 UTR variants in these regions. 2 genes come out. One gene is TGM6 and another one is CENPB. So we confirmed this variant is correct with Sanger sequencing. So then we find this very interesting variant located in TGM6 genes, and we call it R517. This is a mutation, and actually this mutation is occurred in the 2 patients and 6 members of the potential blood cancer patients. Very interesting. It's not occurred in 13 unaffected individuals, and we also checked this variant in a larger population size. We have 500 normal individuals, and we didn't check the mutation there. So we are quite interested to these mutations. So we try to compare with these variants in different animal models. For example, we check the human, chimpanzee, and we found this allele is quite conserved. So we think it must be some function for these mutations. And I conduct a bioinformatics prediction. What exactly this gene, this variant plays? So I find this variant actually located in the beta translin, these regions. And from the bioinformatics analysis, I can know that if this mutation occurred, so this is actually called the GDP binding pocket. Mutation occurred, and this protein will have a stronger binding ability to combine with the GDP. And then and also because this is located in the loop region, so in this region. So this mutation will make this protein become an active form. Active form is the longer form. This is a non-active form, is a little short. So this mutation actually plays very important functions from the bioinformatic analysis. So we finished this project, and we found this very interesting gene, TGM6. And later, there are another publication just use lots of biology experiments to confirm the function for this gene, and now we know that it's definitely a blood cancer gene. So we've submitted in 2005, and EGHG accepted this project. So later I conduct another project called gene-based recessive diplotype exome scanning, and we identified FGF6 gene as a hemochromatosis gene. So this project is exactly what I conduct here. So with the PMRP data, we find if the traditional GWAS study just use a loci by loci approach, but actually if you use a compound hierarchical approach, you can find more recessive genes, and exactly is what we've done. In the PMRP, we find 18 hemochromatosis disease. This disease is a recessive disease. So with exome array data, we identify 2 genes, very significant. One gene is HIV. HIV is already previously reported. But another gene, FGF6, is a novel gene. So we want to know whether this gene is some functional in this disease, hemochromatosis. So we checked the QQ plot, and we found it's quite significant. And we found this gene have 2 names. One name is fibroblast growth factor 6. Another name is heparin binding growth factor 6. The interesting is we know heparin is a very important factor in the RO regulation. So we are thinking maybe this gene will play some role in this disease. So then we conduct a comparative genomic analysis, and we combine all the RO metabolism genes, and we check what's the relationship between the evolution. We found all the RO genes actually have 2-wave evolution. One wave is the first stage. All these genes occurred very early, and from the Drosophila to human. And all these genes, very important, is RO fundamental genes. And the second phase actually is RO regulatory genes, and FGF6 exactly occurred in this region. So we guess maybe FGF6 plays some roles in the RO regulation. So then from the domain analysis, we can predict what exactly this variance is and what are the function for this variance. So we found we have 3 variants in our data. We have the first one is this one. So these 2 variants actually located in FGFR binding region and heparin binding site nearby this region. We are thinking because this variant is occurring in 2 very important domains, so I think maybe they are very important. The third one is part of it is occurring in the heparin binding site. You can think if the variant occurred here, so the FGF6 binding with heparin will be affected. So then we already found the gene and the variants, but we need to make sure if these variants actually have the function. So we use several molecular, cellular, and some other methods to validate the FGF6 functions. For example, the first one is we thinking if we treat the cells with additive protein, what happens for the cell? And we found that if we treat it with FGF6, the ROS in the cell is significantly decreased. Similar, and we want to know what exactly FGF6 influenced. We checked different RO metabolism genes, and we found the HMP gene was significantly increased after we treat with FGF6 protein. And now we know HMP is a very important gene. We can see it's the #1 RO regulatory gene. So now with this result, we totally confirmed this gene is played a very important role in the hemochromatosis. But if you want to publish your journal, you need to make the full story. So we try to insert different mutations in the cells and check what's the factor for this variant. And you can find if you insert the raw FGF6 plasmid, and you can find HMP is a significant increase. But with mutation plasmid, you can find HMP don't have very strong response. That means if the mutation occurs, then HMP cannot be increased. So similar, we use a Western plot, and we check some other disease, for example, the systemic sclerosis and the liver cancer. We know this disease, the iron have some abnormal. So we notice a similar result. For this disease, the FGF6 was downregulated, and we noticed the iron was significantly increased for the disease. So eventually, we give us a novel model how FGF6 regulate iron metabolism. So in the normal situation, FGF6 can combine with FGF6 receptor, and then this pathway will be open, and then HMP gene will be increased. Increased HMP can replace the ferroprotein protein, and then irons cannot be released to the human body. So for the normal individual, we have the normal FGF6, we have the normal heparin, and then we will have less irons. But if FGF6 was broken, so we don't have HMP, and then the repressing function was broken, so lots of irons will be absorbed by our human body. So that's why hemochromatosis patient have more irons in his body. So this two examples showed how to identify disease genes with genetic analysis. But the question is, lots of disease, especially for complex disease, epigenetics only contribute I cannot say very limited, but just maybe 20% or maybe 30%. But the majority, a lot of contribution maybe from the epigenetic environment, lots of this interaction. So how about we just use some epigenetic biomarkers to do something? So in this study, I have 4 different projects. The first one, I'll give you some background. So human genome is not linear. Actually, we have different modifications. For example, in DNA, we have demethylation, DNA hydromethylation, lots of some other modifications. For histone, we have a bunch of, maybe thousands of different modifications. But it's very hard to do that because there are too much these different modifications. We also have another system called non-coding RNA. This is also very interesting topic because this RNA, they don't translate to protein, but they played a very important role to regulate the gene expression. So for me, I spent almost, I think it's almost 10 years to do demethylation research, so I can share you lots of story about demethylation. Demethylation is a very powerful biomarker to do lots of things. For example, we know twins have a 100% identified DNA genome, but if you check the methylation, you will find the twins actually, the genome methylation is not the same. Actually, the correlation is just 0.9. That means we can find lots of different methylation between twins. Also, methylation is very significant associated with the aging. As you become older and older, methylation will change step by step. The most important rules for methylation is it can be used for biomarker to diagnose cancer. You can use the demethylation to separate cancer and normal very, very clearly, actually. So this is the first project I have conducted. All us knows the methylation can be useful for cancer diagnosis, but any individual lab, we just have very limited money. We cannot conduct a very large sample size to get the very accurate result. So for each lab, maybe you have maybe 50 cancer sample, another lab have 100 sample, maybe some very famous lab, they have 200. But even that, the sample size is not enough to make your research go to the clinical side. So how to solve this problem? Very easy way is why not combine this research together? This research, all of them is genome-wide methylation data. And not like the genomic data. Epigenetic data is quite flexible, and you can update to geo database very freely. Nobody will stop you. Not like genetic. Genetic data you cannot share with others. You cannot send it to public data. But the methylation data, actually in geo database, we have a huge number of this data. You can try to combine them together. So then I started a project. How about to find some lung cancer biomarker, combine different studies? So I searched 3 different studies, and each study we have 70 or 30 or 100 samples. But add them together, you can get a very large sample. For example, this project we have the 458 different samples. At that time, this is the largest data set to identify lung cancer biomarker. So it's after you combine this data, because this data is generated from different labs, from different platforms, there are lots of noise between the different data sets. You need to do lots of statistical analysis, adjustment to remove this, we can call it batch effect. So for the batch effect, there are lots of static methods to remove this method. But there's a very interesting way. So we consider additive model, additive effect, and multiple additive effect at the same time. With this way, in my study, I found this is a very good way to remove all the batch effect from different labs. You can see here, this is before the batch effect removed. You can find the largest variation between different labs. It's not cancer or normal. It's actually from different labs. So all these big differences actually generated from different labs. So then, any analysis between, follow this study, we need to remove this fact. So we just use this static method, and I code in this one, and then to try to do that. And you can find, okay, use this adjustment, all the batch effect was removed, and all them looks very shifting. So that you can use this data to generate the following analysis. So then I use some lots of machine learning methods. In this study, I use support vector machine to find the best combination between different markers. I found a very good panel. It's combined by 5 different genes. And with these genes, you can get a AUC value as nearby 0.8. So it's quite good biomarker panel. But if you stop this, it's not okay because it's from genome-wide data. You need to validate your result, give a very solid conclusion. So I collect 150 lung cancer and adjacent normal tissues. And then I want to detect these 5 biomarkers. So I want to what exactly for my early tissues. So this is the result. Because this started in 2012, we don't have a very good method to detect the methylation for 5 biomarkers at the same time. So I'm thinking, can we propose a new method to detect these 5 genes at the same time? Because if you detect them at different time, then you still have batch data. So we proposed, we are thinking, because methylation detection, you can just use bisulfite conversion, translate it to the SNPs. And then you can use the SNP status to infer the methylation. In the SNP analysis, we know we have multiple methods to detect several different SNPs at the same time. So we use this new method. We just transfer the methylation to SNP status, and so that we can detect the methylation for these 5 genes together. And the result is very, very interesting. So all these biomarkers have very significant difference compared with normal. You can see all this is very significant. And also, we have 2 controls. One is the non-difference control. We know that there's not any different methylation in these 2 samples. And as I said, there's not any static difference. Another one is the positive control. This is long one. Long one we know is significantly low methylated in cancer sample, and exactly is what I noticed from my data. So with this data, we can find, okay, single marker have a very good methylation difference between cancer and normal, and the sensitivity, specificity is very good. Usually is nearby is 0.9, but the sensitivity is low. It's okay. It's the common sense because it's for single marker. Usually the sensitivity is not so high. But if you can combine them together, you will find combine them together with some machine learning method. You can find a very good result. So I can show you here. So use different prediction model with these 5 genes, you can find the sensitivity from 0.7 to 0.9. And the species usually very high. This is in the training side. And for the use the model, we observed in the training side, we noticed in the test side, the accuracy is still very good. For example, use the base tree, we found the sensitivity is 0.86 and the specificity is 0.96. It's very high actually. The AUC is, ACC, accuracy is 0.9. So we think use this way we can find some of very interesting biomarker in the diagnosis panels. So summary this prize, this project, we found data integration of the demethylation is helpful to identify useful and solid biomarkers. And the batch effect must be removed before you can take any static analysis. And we propose a new method to detect methylation for multiple loci at the same time. And in this project, we found 2 methods quite powerful for the prediction. Very interesting. This 4 method is what I used in my manuscript. Recently, I redo the data analysis again because recently neural network is very popular. So I want to try whether the new method can get a better result. But interesting, I didn't find this popular method didn't show a very good result. So maybe for this small data set, the neural network didn't get very better performance. So another project is for the liver cancer. So liver cancer is very interesting because we know there's a very interesting risk factor for the liver cancer is HBV infection. And actually, if you can find a lot of HBV infection patients, maybe they will get liver cancer maybe several years later. So we want to know, can we identify some biomarker they are occurred in the very early stage? Because usually when we do the biomarker research, we use cancer samples, but the cancer sample you use, the biomarker is for the cancer, maybe not so useful for the early diagnosis. So we just use this stage and a different sample to check whether we can find some early biomarker. Another thing is we don't want to do the tissues because in the future you cannot use the tissue methylation to the diagnosis. So we know that all the cancers will be died and the DNA will be released to the blood. So can we collect the blood in the cancer patient and for different stages and whether we can find some methylation signal. So okay, so now we put almost 30 different patients together because the DNA is actually very low for this cell-free DNA, and you know this project is started in 2012. So at that time, low level demethylation technique is not so much. So we need to pull them together. After I pull them together, I can use a protein called a methylation binding protein. This protein can capture hypermethylation DNA fragment from the blood or from the urine. So then we can get the hypermethylation, and then we can use next generation sequencing to get this fragment. We can match this race to human genome, and then we'll know, okay, in this blood, what kind of gene will hypermethylated, so that we can get lots of biomarkers. And this is the result of what I will do. Each bar is a hypermethylation in human genome. And in HBV-related hepatitis, you can find a lot of hypermethylation region. But as the patient becomes to dangerous situation, for example, cirrhosis or the liver cancer, you can find the methylation, more and more hypermethylation occur. So if you merge the 3 different data sets, if any biomarker shared by these 3 situations, and we can think maybe they are early biomarkers because they occurred in this stage and they are also exist in different stages. So we just found these 240 biomarkers. We think it can be used as early biomarkers. And we check one of them. You can find, okay, from the early stage to the liver cancer, the methylation is increased step by step. And also we check the gene expression in the tissues. And we found as the stage go on, the expression becomes lower and lower. So that means this methylation may be a functional methylation loci. So with this method, we can get we can compare the sensitivity, specificity for different markers, and we identify some very interesting biomarker. The AUC is 0.7 to 0. Nearby the 0.7. So in this study, we validated 3, but actually there are lots of interesting biomarkers. In the later study, we can have some further research. So all the data actually, I updated this data to public data. Anyone who are interested in this can download it and make a further analysis. Another one is blood cancer. We know blood cancer is quite interesting because as the tumor grows, so blood cancer will die and the DNA of the cells will be released with the urine. So you can think we can collect the urine and collect the DNA, then we can find some diagnosis approach to infer the blood cancer. So with this idea, we want to find some biomarker for the blood cancer. Then the first step is we find some blood cancer cell line and normal blood. Then we we use the method-seq. So the method is same with last one. We just use protein to capture all the hypermethylation region from the cancer samples. This one is from the cancer cells. So step by step, this all is bioinformatics that I don't want to talk too much, but eventually we find thousands of these very interesting biomarkers. So thousands is too much for us to make the follow research, so we need to scale it down. So for these 1,000 markers, we are actually looking at 100 genes, and with some samples, we scale down these genes to 49, but we still find this too much, too many. So we just use another sample. So okay, eventually we find very powerful 9 biomarkers. These 9 biomarkers, we just increase the sample size. This is a very large sample size at that time. So we collect totally 400 urine samples. 100 normal urine, 200 urine from the blood cancer, and some other blood disease. This is what we found. For these 9 biomarkers, the sensitivity is about 40, 30, something like this. Some one is bad, but some one is powerful. This one, the sensitivity is 42. And specificity is usually high because it's very easy to find some cancer hypermethylation, but for the normal, usually it's very unique. So eventually for cancer, a single marker sensitivity is quite a long range. If you use multiple biomarkers, the sensitivity is high. It's usually 70% or higher than that. And the AUC is 80%. So we identified this biomarker and this panel, and we didn't stop. We want to check whether we can use it in the clinical site. So this is what we done. So we recruit 48 suspected blood cancer patients, and we use the traditional way. This is the golden standard to know whether this is blood cancer or not. So these 48 patients, there are 33 is blood cancer, and 16 is not. So we use our methylation panel to identify, and we found. So 25 of them is blood cancer. So the sensitivity is 78, and 14 in 60 is negative. So the methylation signal is also negative. So specificity is 87. Very interesting is you find here there are 2 normal individuals. This shows a positive methylation signal in our panel, right? So you can take this as the false positive, but actually we think because methylation signal is very good early biomarker. So we think whether these 2 patients maybe get cancer because the methylation is already positive. So we followed 2 years for these 2 guys, and they're very interesting. One of them had blood cancer within 24 months. So we think maybe methylation-based panel can identify some false positive result from the golden status. Another one is we want to know whether methylation assay can be used to assess the surgery success ratio. So this is the idea. So we have 26 confirmed blood cancer patients, and then we use our methylation panel detected this patient, and we found 21 urines show the positive methylation signal, and we check this this 21, because this 21 patient will be accept surgery, so they will remove the cancer. So after remove, we found that 19 of them, the methylation signal disappeared. So that means cancer cells were removed. But very interesting, there are another 2 individuals, even you accept the surgery, there are still methylation signals there. So we're thinking maybe the cancer didn't remove completely, so that the cancer cells still release to the urine. And then what the pity is, we followed for about 1 year, and then these 2 individuals lost. So we cannot give you the answer here, but I think it's a very interesting idea to use methylation panel to evaluate surgery success ratio. So the third one is what is the most interesting one. If you know it's blood cancer, then here already blood cancer. The question is lots of individuals, they come to the hospital, they don't know what cancer they get, or they don't know whether they get a cancer. Even in the early stage of the cancer, the symptom is not so clear. It's not obvious. So that's why majority of patients diagnose cancer at a very late stage. So the question is, suppose some cancer DNA methylation was released to the blood, and we do find some signal. Can we infer which tissue will release this DNA? Actually, we can do that. Why? Because the tissue have different methylation platform profile. So use this different profile. Actually, you can match this fragment to different tissue. For example, from liver or lung or what. So then we are thinking why? Because this idea is good. Why there's no any publication before, right? People are smart. Why they didn't find this? So we checked the previous measurement for the demethylation, and we found majority they use 3 different methylation measures. For example, methylation fragment, methylation entropy, or epipolymorphism. You can find this is 5 different methylation profile. Actually, this profile is occurred in the human blood. So this is totally unmethylated. This is totally methylated. But this one is okay because it's very easy to do. But for majority situation, there may be this 3 situation. This one is 50% of the their fragment is methylated. Another 50 is non-methylated. This one is totally heterodyactyl. This one is the first 2 is methylated. Another 2 is non-methylated. This is quite reverse. So use all these 3 measurements. You can find you cannot tell the difference between all of them. For example, you use methylation or low frequency. This is the zero. This is 100% methylation. All of the 3 situations is 50% methylation. For this method, this is zero entropy, but this one have the same entropy. So there's not any measurement that can tell all the different patterns for this situation. So we think maybe this is a reason why we didn't achieve this approach. So we're thinking why we propose a new pixel. We call the methylation habitat load. And this is the formula for our method. And we found this method is quite good because it can tell the different, it will have the different value for different situations. This 0, 1, 0.5, 0.16, and 0.120. Any different situation will have a different value so that we can tell all the difference between them. Oh my God. So sorry. So we need to fast. So then we use this new concept to find lots of biomarkers for the methylation habitat load, and we found lots of biomarkers to separate the different tissues. And then we do the non-supervised method to identify what kinds of biomarker will be interesting. So use non-supervised method, we find this group will be very interesting. They are low in all the blood, all the other tissues, but higher in the cancers. And this DNA actually will release to the blood. And actually what we found, okay, for these regions, for the colon cancer plasma, we will have very high value, but for the normal plasma, we don't have this value. We also use that method. We proved our method have a higher power than the previous method. So then we build a prediction model, and we find use these tissue-specific biomarkers. And if you get the blood from colon cancer, lung cancer, and then you can mapping this plasma to its own tissues. This is a very interesting result. And we found the sensitivity specificity is okay, not so bad, because this sample size is not so large, but we think this result is okay. And this result was accepted in Epigenetics in 2017. So okay, I finished all this project, what I have done. And very interesting is what I'm going to do here. This is, I think, most of you will be interested. So I have two proposals. One proposal is contribution of the genetic variance in epigenetic factor to human disease. Why I conduct this study? Because I have got a very good genetic training and I have a huge number of epigenetic research. If you combine them together, I have an advantage compared with others. So I just write a proposal and submitted it to the CRBN last year, and very interesting. I was accepted by this program, and they think this idea is good. And other one is my expertise. I want to do it cell-free DNA with the pan-cancer early scanning in multiple clinic. So I have conducted different cancers, and I know how to map the DNA fragment to the human tissue. This is just what I list. You can check my other publication. I have lots of some other details. So already I finished all these individual cancers. Why not we can go to the pan cancer? Actually, from my experience, I checked lots of cancer data. They share lots of common biomarkers. So not only this, my skills is bioinformatics and epigenetics. I think much clinical research into it is quite required with these two skills. And I can not only these two, I can collaborate with Dr. Fried on the epigenetic profile of eye disease, or Dr. Carville on the heart disease, some other with Dr. Harbin, and we can investigate the epigenetic longevity, methylation clock, or Sherman's knots. Dr. Shukla, I can do together with him about epigenetic changes in multiple-induced disease, just like HBV in cancer. So I have a long time work together with him. I can work together with him to propose some new approach to identify this gene. With Dr. Hardy, I can do some epigenetics initiatives in the birth defect disease. So actually, these skills, I don't have any overlap with other my colleagues, but I can contribute my skills on the bioinformatics and epigenetics so that we can have a whole story for different disease. So this is an interesting thing. So for the different, we have so many different. If you check the genetic heritability, you will find the heritability is not so high. Usually you're less than 0.3. But if you can find the shared environment effects is very large. This is actually the interaction between the epigenetic and the genetics, because the environment cannot change your genome, but the environment can easily change your epigenetics. Okay, then the question is, okay, we know that epigenetics plays so important roles. How about this in epigenetic factors? In human genome, we have our 250 epigenetic factors, for example. Some protein were used to modify DNA. Some protein were used for histone. So the genetic variants occurred in this epigenetic factor will play a very, very important role, right? So then I want to answer the question. In KMRP, we have about 2,000 different phenotypes. So can we find which phenotype have more collaboration with the genetic variants in this epigenetic factor? And then we can answer, okay, maybe this will have a significant collaboration with epigenetic research. So I have a very good pipeline how to do that. How to identify epigenetic factors, so the origins, how to identify genes, how to cover preliminary data, and how to conduct the methylation research to the PMRP data, and how, because majority of my work is machine learning, deep learning. I need to build the machine learning method, build the prediction model, and build our package. So this is the whole story what I proposed in CRBM project. So this is preliminary data. I only have 15 phenotypes because some other phenotypes need to get the RB approved. So for these 15 phenotypes in PMRP, and for all the 250 genetics factors, and we found some very interesting signals. We found 1,017 signals, and for different diseases, you can find some gene will jump out, and we can find some phenotype will have more collaboration with these epigenetic factors. Not all you can find some other gene, maybe they don't have so many significant signals. So this is the first one. Another data is M3. I can, because I already found these two SNPs, so that I can enrich this SNP to the pathway, and then I can infer. I found lots of SNPs, and then I can do some public data from, for example, the expression data or the gene regulation data, and to mapping where these variants play roles. For example, these variants only play roles in the liver or the lung, so that we can match these variants to different tissues, and then we can get some function for these variants. So another project is cell-free DNA research for pan cancer. Pan cancer is, I think it should be very, I think in the next 10 years, this will be very popular even in hospital. Because the method is watched and the series is very good. So for this project, my aim is first one. So identify pan cancer demethylation biomarkers with deep learning algorithm. The second one is validate this biomarker in multiple clinic systems. The third one is test the performance methylation pathway in cancer risk new carriers, because this one can be used to actually real prediction. So all this work actually can be conducted very quickly, because I already collect all the data, the TCT pattern data. In the data we have 10,000 RNC data, 20,000 demethylation data, and 80,000 mutation data. All this data is for sim samples. So we can infer lots of things, especially we can identify lots of very interesting pan cancer methylation biomarkers. So we have 33 different cancer types, and 1,000 normal, 10,000 cancers, and 90% of them have the overall survival time. So we can use the survival analysis to demethylation. And also all this public data is already collected by me, and all this is saved in our computer. So this machine learning method, deep learning method, the platform is already done for me. Actually, you can see I have lots of this data. So usually, actually, all this works. Some of them is already on the middle, on the way. Some of them can be started very quickly. So let me show some preliminary results for this result. So in order to get a very accurate result, because we have 10,000 cancer samples, but not all of them have the shared normal. Some of them only have cancer, don't have the normal, or the adjacent normal. So if I want to get accurate results, so I just select the case. We have the cancer tissue, and also we have the adjacent normal tissue, so that I know whether my analysis is good or not. So if you check the genome methylation data for all these cancers, and you can find, okay, so similar cancer always class together, and similar normal we always class together. That means your methylation is quite a good marker to identify the different cell type or different tissues. And if you compare the correlation between different tissues, then you can see, okay, so this is lung cancer. Lung cancer have two. One is adenoma cancer, another one is squamous cancer. So you can find that for this cancer, actually, they have very high, they have a little high correlation. But if you compare with normal tissue, you can find, okay, the correlation was decreased. That means the cancer have higher heterogeneity compared with normal tissues. So similar thing with the two different kidney cancer. Kidney can also have some subtype. And if you check the kidney in normal tissue, you have very high correlation. The correlation is 0.87, but for the cancer, you can find the correlation is decreased. So we have the different cancers. So can we predict the different cancers with genome methylation data? How many biomarkers we need? This is very interesting, because if you need 1,000 biomarkers, you can never make it in the hospital. So I just use one of the machine learning methods, random forest, and you can find here, very interesting. You only need, as the sample size increase, as the feature size actually is biomarker. As the biomarker increase, you can make the accuracies become higher and higher to predict different cancers. And after the 25, the accuracy actually becomes stable. And if you use 75 biomarkers, actually the later you can find the accuracy become very stable. You tell us for 11 cancers, you only need 12 to 17 and you can tell exactly what cancer it is. It's lung cancer or liver cancer. This is the first step for pan-cancer research. The interesting is you can find here, this is lung cancer. The accuracy is very low because the lung cancer has very limited sample size. In the level. For this one, it only have 26. Lung cancer is only have 26. So sample size is too less, so we don't have very good result. But if you use all the samples, this is a peer sample, right? If you use all the samples, you can find the accuracy is quite good. And the AUC is nearby 0.9. And I'll show you one example why you can find this result so good. Because you can see here, this is a one biomarker. This is colon cancer, blood cancer, brain cancer, breast cancer, liver cancer. You can find for this biomarker, they are all hypermethylated in these different cancers. So actually, go over all the human genome, you can find lots of this interesting result. So another thing is, this is pan cancer. It's very easy to get one individual cancer. This is GBM cancer. This is brain cancer. So for brain cancer, for the TCG project, we have about 500 samples. But you can collect more samples from GBM. So for this study, I think it should be larger sample size. I collect 1,000 brain cancer samples and 1,000 normal control brains. And use these samples, I can add into here. You can find this is one cancer type, this is another cancer type, this is normal brain. So they are clearly separate from different cancer. And this is different result. You can see here. This samples are separate from this is a female, male. You can see there's no any difference between male and female, right? So this is a non-supervised class analysis, and you can find here, this region actually is pan-cancer biomarkers can be used in different cancer types. QQ plot shows we identify lots of interesting biomarkers, and then you can try to attack these biomarkers in brain cancer. We have the survival data, so we can conduct a survival analysis, and actually I identified almost 600 different translation sites that are related to the overall survival time. So this is what I have done. Okay, so I think the time is almost done. So I need to say thanks to all my supervisors. This is my PT supervisor, Dr. Chiu is in the University of Texas, and I was in his lab for 2 years, and missioning to a computer analysis. Using the software, something like this. Dr. John is working in the UCSD, and I conduct self-reading research and twins methylation research, and another is CRISPR-Cas9, because CRISPR-Cas9 is very popular, but the static method in that one is terrible. The data is very dirty actually. So we need lots of static method in CRISPR-Cas9 study. So then it's even truly I work very heavily here. That's really I think in his level I have 3 or 4 different projects. Yeah, everyone looks quite good. So I should say thanks to all of you. So and then thank you so much for all of you to coming to listen my presentation. Yeah, thank you.
First of all, excellent talk. A lot of great work you have done. 2 questions. One is specific question. Your gene of iron metabolism, I believe it was FEF6 gene. Many pathogens rely on iron for their virulence. Did you see any correlation with that group has increased infections or anything? If I understood correctly, that you found the gene.
Yes.
In that cohort of patients, was there any increased bacterial infection? Because many bacteria rely on iron for virulence. Because it looks like, it produces abundance of iron in blood.
Okay.
If I understood it correctly.
So yeah, this is interesting question. We think, we use that, right?
No, we didn't look at that part of their medical record.
Okay.
Because so you mean last time I gave you the solution, we want to find that this subtype of hemoglobin we think is want to prove this setup is machine clinic type. We want to do that, but we didn't have the clinical information, so we cannot claim that. So it's very interesting, actually, to discuss with you. We want to do that.
Sure. Second question, you're looking for the markers. How early or how many years before the actual cancer you should have the DNA where you can look for the methylation pattern so they can predict there? In your opinion, based on your research, like at a particular age in life or, how many years before the disease, you can still have markers that you can predict.
So according to my experience, the methylation will be earlier than the symptoms is 2 years. So 2 years, you get the DNA methylation, and then waiting for 2 years, you can get some result. But one thing is not good news is cell-free DNA is stable, but not so stable. Usually you can keep it for 6 months to 8 months. So that means, so if you use cell-free DNA, you can only warning the patient 8 months ahead of the symptoms.
Thank you.
There's a gap between the start of the cancer and the cell-free DNA can warn you.
Okay. Thank you.
So maybe I'm misremembering. I know that there are histone deacetylase and histone demethylase drugs. Are there DNA demethylase drugs? And if so, have you looked at the methylation profiles of people who've been treated with them in the past?
Yeah, that drug is used to blood cancer. It's actually demethylation because for the cancer, the methylation genes is tumor suppressive gene. So tumor suppressive gene was silenced and then were hypermethylated, so gene expression was down. So that drug was used to remove this demethylation on the tumor suppressive genes. This is what it works. But actually, genome-wide level, DNA is downregulated. Demethylation is downregulated because in our human genome we have 60% of the genome is a repeat region. This repeat region should be highly methylated because we don't want this repeat gene transcript. And for the cancer, it's not. They just remove the methylation on this repeat region. So this repeat elements actually jump in human genome all around. So yeah, this is a very interesting thing for methylation. And actually for some virus, for example, HBV, they just go to insert their human genome, their DNA into the human genome. And they will carry some DNA sequences to go outside the human genome. And the methylation will change. So that means, I guess maybe that's why HBV can induce the liver cancer later. But it's just my hypothesis.
Yeah, I mean, the naive hypothesis for the increasing methylation with increasing age correlation would be that conceivably DNA demethylase broad treatment might actually reverse some of that. I just don't know if it's ever been investigated.
So it's a very interesting field. Not so much data outside. It's just becoming very hot from 2013 something like this, because the cell-free DNA extraction and sequencing technique become more. So if you search the data outside, especially for public data, not so much. So we need sometimes we need to generate some data by yourself. And I know some labs they don't want to share the data to others. So just want to get more information from the data.
Just a trivial question. What proportion of cell-free DNA in a patient with cancer is cancer DNA versus wild-type normal DNA?
So, from the estimation, usually it's 5% of the DNA is from cancer. 95% of DNA is from the
DNA or cell-free DNA?
cell-free DNA.
Okay.
Cell-free DNA. So, but there's some difference. For some very advanced cancer patient, this value will be very high. Maybe 20% of DNA is from cancer, especially for the stage 4. In the early stage, you cannot find so much. You just need maybe 5% to 10%. 10% is actually very high. Usually 5% or less than that.
So you need to have deep reads to be able to differentiate then, I assume, the baseline versus tumor?
Yeah, you need a deep rate because suppose it's 5%, right? So you need at least 100 days so that you can get 5 cancer DNA fragment. So, but the interesting thing is, cell-free DNA is very actually, for each ml, how many cell-free DNA is just 10 nanogram, right? For one ml, only 10 to 20 or a little higher, 50 nanogram DNA. So this DNA actually cannot give you very high, actually. So you just sequencing all of that, and then you can pick up there's some 5% of it from cancer from this normal. Even you give very high coverage, it will be just a repeat. Because the DNA is very limited, you can get full coverage for this one.
Can you sequence the genome with cell-free DNA? So if you have 100-fold coverage, you're going to have chunks across the whole genome, presumably, unless if there's a bias between what's actually circulating. A section of chromosome 20 is only circulating versus a chromosome 1. Just kind of curious about what the limitations of the assay is and what it provides.
This is a challenge. So one way to solve your problem is don't do the genome-wide cell-free DNA method. You just use, for example, RBS. RBS is use some enzyme to cut the human genome to remove, for example, repeatedly, only focus on gene region. So that you use this small region because gene only count 5% of human genome, right? So you use this 5% of human genome, you can get a very high coverage. Or else you just focus on the repeat region, so you can use, as I mentioned to you, LAN1 or SAN1, use this kind of one, you can get very high coverage. So when you pick up a technique, you can try this. For the methylation, there are at least 20 different approach for you. If you want to cover 100 genes, or 1,000 genes, or 10,000 genes, or all genes, they have different methods.
There's a population of immune cells that excrete their genome when they're angry. It strikes me as that sort of thing that might stochastically make a cell-free DNA assay fail, right? That if there's a certain kind of infection in the wrong place, and now you're dealing with 1,000 times more cell-free DNA as a result. It seems like that would be an issue.
RNA is very significant. Even you give a medication, you are nervous, or even you are sad, RNA will be changed. But the methylation is not so significant. It's a little sensitive, but not like mRNA. So because if you want to change your methylation, you need to go to the next generation of the cell. So for the first generation cell, you want to change the methylation, you need to get another generation. Not like mRNA. mRNA you can change any cell, even you don't go to the next generation. So methylation actually is flexible, but not so flexible. Currently, according to my data analysis experiments, cancer is very strong. Smoking, we think it's very strong, but if you check the methylation data, it's not so strong. It's strong, you can identify thousands of these markers, but just thousands.
Yeah, I guess what I was trying to say is that if there was an infection triggering that particular immune response, the cell-free DNA as a proportion of the total cell-free DNA from the tumor would go down possibly several orders of magnitude, because the bulk of the circulating DNA would then be derived from the immune system and not from the tumor. But I don't know how common that is. I just know there is a population that does that. Have you seen that? Have you seen people, data points where it'll suddenly just completely drop down and then pick back up at the next data point, or?
There recently data come out is through the exercise. So they take whether you take a very strong exercise, it will change the cell-free DNA chain. I mentioned this even before. So they do find the cell-free DNA level change. I mean just level change. Level change doesn't mean anything. So level change, you can only show, okay, some cell maybe death, right? So DNA will release. But the very interesting thing is the methylation is tissue specific. So liver cell death or the lung cell death, they release the DNA to the blood. This is different. So level is one side, another side is the PET different situations, I think we need to check the data and then get a conclusion.
Last question.
Have you been able to, or have you used your methylation technique to identify like tumors of unknown origin? So there are sometimes primary tumors that pathologists and oncologists can't actually diagnose. Could you potentially use your pan-cancer technique to then identify what tissue of origin those tumors generated? Or in your technique were you just define using known tumors or tumors that were blinded and then positively predicting where they come from?
For this situation, as first one, we first need to identify some pan-cancer biomarker.
Right.
Second one is we need some tissue-specific methylation signal there, or else how you can match it back, right? So we need two signals. And for the second one, it's a little hard because methylation data is not so large. We know we have for example, for the lung tissue, liver tissue, we have the reference for methylation. But for majority of tissue, we don't have the reference. So there will be a little difficulty when you match it. Maybe it's from kidney. We don't have kidney reference, so maybe it's a little hard to match it to there. So currently, I can tell you that find the pan-cancer biomarker and try to use exactly tissue-specific marker to make the prediction. If you want to very much production, I think lots of sense to be seen.
Okay, and I guess the second question is, could you do something similar? Have you tested looking at primary versus metastatic? So looking at those two different tumors from the same patient,
Yeah.
and obviously there are known differences and there are changes in
There
somatic
should be
mutations.
some difference.
But
Because
you'd expect to see differences, but have you tested that?
not yet. It should be why because the evolution theory of the cancer cell, right?
Mhm.
So the cancer cells just evolution to different direction, right? So one direction maybe go to another side, they will get a second cancer, right? So you can think during this evolution, the genomics change. So I think epigenetics also will change. If you compare whether they are different, I can tell you they are different. But if you ask me, can you get some biomarkers? They are shared.